Namaste all of you. My name is Dr. Ritu Singh Rajput, Faculty of Agriculture and Veterinary Science. Today we will discuss about the equilibrium dialysis. So, equilibrium dialysis is the specific application of the dialysis that is important to the study of the binding of the small molecules and the ions by the proteins. It is one of the several methods available for the purpose, and its attractive feature continues the physical simplicity. Another attractive feature of the equilibrium dialysis is the ability to perform interaction study without use of fluorescing and the radio label tanks. How does the equilibrium dialysis work? First of all, you have to take you have to before the dialysis we have to see the non-binding ligand, the sample chambers, and there are two types of chamber between the particular membrane, the sample chamber and the SI chamber. And after filtration, if the ligand and the protein do not bind to the each other, the ligand is the free to cross the membrane. At the equilibrium, the concentration of the ligand in the SI chamber will be exactly half the initial place in the sample chambers. So if they cross the membrane, they can easily protein and the ligand bind the complexes. After the dialysis, binding ligand, the sample chambers, the ligand and the protein from the complex, the bound ligand will be unable to diffuse across the membrane and remain its sample chamber. The concentration of the ligand will be still equivalent on the either side of the membrane upon reaching the equilibrium. In the cases, however, the ligand concentration is the SI chamber is reduced by the total amount of ligands bound to the protein divided by the two, which is unbound ligand and the two types of ligand, binding or non-binding ligands. Generally, the equivalent dialysis is the measured amount of ligand bound to the macromolecules, and is typically done by the indirectly process. In the mixture, ligand and the macromolecules are difficult to distinguish between the bound and the free ligand. So, free ligand can be dialyzed through the membrane until its concentration across to the membrane at the equilibrium. The free ligand concentration can be measured easily, and you can obtain the properly data after the binding parameters and the compounds such as the binding constraints and the number of binding sites, and it will be depend on the number of binding sites and the binding capacity. That means that is called as the affinity. If the protein has particular affinity to bind the particular ligand, so they can be bind